The Babraham Institute undertakes world-leading research into understanding the biology of how our bodies work, including what changes as we age and during disease. In Scientist Stories, we invite you to meet some of the scientists behind the research and discover more about what they do and how they got to be here.
In this latest Scientist Stories, we're talking to Claudia Ribeiro de Almeida, who is a researcher at the Babraham Institute. So can we start, Claudia, with you telling us a little bit about your role at the Babraham Institute?
So I joined the Babraham Institute 2 years ago, a little bit more than 2 years ago, and I'm a junior group leader. I've just started as a tenure track group leader and more recently as a Sarah Riddell fellow.
Can you tell me a little bit about your area of research?
So our group focused very much on the molecular immunology question. So we want to understand how B lymphocytes generate a diverse antibody response. And B cells have this amazing mechanism to deal with this problem, which is having a varied antibody response to fight almost or virtually any pathogen that they can encounter. And at the molecular level, this is achieved by cutting DNA by a mechanism of DNA cut and paste. So antibody genes are encoded in very small gene segments that need to be reassembled before you form a gene locus. And this occurs by a mechanism of DNA cut and paste and occurs in a random manner. So this mechanism has been studied for a very long time, and we understand it with a great level of detail. So we know that the locus needs to be transcribed in order to open up and initiate recombination. What we didn't know so well is whether the RNA transcripts that are generated via transcription have a role. And more recently, it was shown that these transcripts can fold in particular structures and this direct recombination to antibody genes. And our work is very much focused on the role of these RNA structures and RNA binding proteins that remodel these structures, known as RNA helicases. That's what we are studying at the moment.
Can you tell me a little bit about your background and how you got to where you are today?
So I'm originally from Portugal, and I studied in Lisbon. There was, at the time I joined university, there was a little moment of doubt where I was undecided whether I should follow medicine or biology. I eventually started in biology, which is something I don't regret because I really loved it. And I think it was purely because during my secondary school years, I really enjoyed studying biology, and I just wanted to know more about the living world. So I did biology in Portugal, and then at the end of university, I moved to the Netherlands to do a PhD, and I spent 5 years in the Netherlands doing a PhD very much in the area that I'm working now, so in molecular biology, antibody gene recombination. And it was an amazing time, both personally and also professionally. I really enjoyed living abroad, getting to know about another culture, another way of working. And I think it was very important for myself and my career that time in the Netherlands. And then I moved to the UK. When I started my postdoc, I did a few years of postdoc in London. And then I moved to Oxford, where I spent a bit more than 6 years before moving to the Babraham. So there has been a lot of mobility between different countries and cities, but I've been really enjoying that aspect as well.
Yes, I think that's something that is often a positive of working in science, is that you do have these opportunities for travel and exposure to people from all sorts of different countries and cultures. And I think that's a great joy for those of us from the UK, in the UK, being part of this big melting pot actually of people from other places.
Yes, and the UK is great because of that as well. I really enjoy the fact that you know people from all around the globe. It's really a privilege to experience that, and it's it's a challenge as well to work with so many with people that have so many different cultural backgrounds, but in that challenge there is great value, I think.
Can you tell us a little bit about your other interests outside of science?
At the moment, well, life is very different from what it has been in the past. One thing I particularly enjoyed in this past year, when we were mostly in lockdown, was to cook. I think partially because not being able to go to the lab, it kind of distracted me and made me feel a little bit, I think cooking is a little bit like experimenting in the lab as well. So I enjoyed cooking a lot. I tried to master all the traditional Portuguese dishes, which are really laborious and take much time. So whenever I have some energy and time, I like to spend some time in the kitchen.
Can you tell us a little bit about what a typical day looks like for you?
So I would say that one big change at the transition from a postdoc to a group leader position is that you are not longer able to fully dedicate your time into bench work, doing experiments, which is something I really like. So nowadays, I try to be in the lab a little bit and do some bench work, but on top of that, there is a lot of meetings with my lab members to discuss experiments, discuss their results, and planning experiments ahead. There's a lot of reading as well, and a lot of writing. So there's a multitude, and there's also a lot of more housekeeping type of work, so things that we need to do in order to keep the lab running. So we are still a very small team, and part of those things are still very much taken care by myself.
What advice would you give for people looking to get into science?
Mm-hmm. I think nowadays there's a great deal of information, and the UK is amazing in public engagement. I think there are plenty of opportunities to get to know what and also to experience in the lab what it is like being a scientist, what it is like to work in the lab and doing lab work. I think it's just trying to get in touch with research institutes and trying to gain experience in a lab setting as early as possible. And I think, at the BIBRAM, we do provide those opportunities, which I think it's really important for the students. And that should give you a very real understanding of what it is like and which aspects of the work you like, because there is the theoretical part. I really enjoyed studying biology, knowing all about that, but in the end of the day, your day-to-day job encompasses many other things that you also need to know if you like. And I would also say that nowadays, there's also many different types of scientists. So you can be more lab-based, or you can be a more computational scientist. So it's really variable, it's really flexible. So it's good to understand that and see if you can see yourself doing those many different things.
What are the real-world implications of your research?
Obviously, understanding how an antibody immune response is generated is important, because it's part of our adaptive immune system, and it's also the basis of proper response to vaccination. It's interesting to understand the role of new proteins involved in these mechanisms, because this would allow us to tackle this process when it occurs with errors, like, for example, during autoimmunity or during the development of B-cell lymphoma. And in particular, RNA ligases are very interesting drug targets. So our ultimate goal is to be able to identify RNA ligases that are implicated in this mechanism, and probably by doing that, understanding why sometimes this mechanism doesn't go according to plan, and be able to therapeutically target it.
What do you love about working in science?
Yes, I think it's the fact that you can, you can look into what is known about something, but there is a big amount of freedom that you have in thinking and in creatively thinking how things can work. And for me, I think it's the creativity associated with science that I really enjoy. And I think that has been a little bit of a common theme. So, working on antibody gene rearrangements, it's an area, it's a very intensive area of study. There is many groups around the globe studying these mechanisms, but they're still, appreciating that there are still things we don't know about it, and implicating new factors and discovering or proposing ideas how these new factors can be working. I find it really exciting. So I think it's the creative part of the job that I really like.
What might your next steps be?
Yeah, so absolutely, yeah. I think at reaching this stage, obtaining your first position as a group leader, setting up a lab, writing your fellowship, getting the money, it is really a process. And although I have only started or already started 2 years ago, it does feel very much like the future is now. There was there was a lot of planning involved, and I'm just looking forward to get those ideas to work in the lab, to establish my team, consolidate our research, and building my team. So we are still a very small group. I will be able to recruit another postdoc soon, and hopefully in the next 3-5 years, I would be able to recruit a few more people, grow a little bit more. But the future as I plan it and envisage it up until now, it's very much happening now. So I'm very much looking forward to just get done with the job and enjoying it.